I'm actually Julie Holder. I'm representing Roslin Cells Sciences, which is actually the sort of coordinator of, in collaboration with Tim's role in actually coordinating the EBiSC consortia. And I'm going to talk about building the European iPSC collection to really meet your needs, the researchers' needs, and on demand. So, what my talk is going to actually be on, the overview, the introduction, and the initial hot starts. And we actually really started this collection with a series of well-established iPSC lines that were available from academic collaborators to really generate the data to allow us to make some of the decisions. Some of the information that I will be giving you is actually on this hot start and how we've actually really interrogated these iPSC lines and learnt an awful lot from them. In addition, I'm going to talk through some very brief summaries on how we use these cells and then summarise how you can actually obtain them, which will be continued throughout this talk this morning. So it's a consortia consisting of a very large amount of partners, and we all integrate together, actually working with each other to facilitate the requirements for depositing the iPSC lines and really expanding and utilising those iPSC lines in differentiation assays to different lineages. So to build the catalogue, as I said, we had these 7 iPSC centres that really contributed to the hot start lines. Underlying was the Sanger ISSCR lines, which are of normal origin. And in addition, the FPIR partners actually commissioned particular lines of interest. So we've done a lot of gene editing with Bioneer, and this is something that there are a number of posters on throughout this ISSCR meeting. In addition, we actually have utilised some IPS lines from other EU-funded projects, and the makeup of these lines has really fallen into some very discrete areas, mostly neurodegenerative and psychiatric, cardiovascular, some minor genetic variants, metabolic such as type 2 diabetes, blood and autoimmunity, and musculoskeletal disorders. The actual continuum at the bottom on that of this slide actually shows the cell line receipt from the iPSC derivation center, and then the foring, we actually register it into ISSCR, and we cryostore a number of particular vials to establish that sort of master cell bank. We then for the cell lines into various plates and culture them on in a feeder-free environment with no antibiotics, and continue that throughout the expansion process. So we've had experiences of cell lines that have recovered well or haven't actually recovered well, and then we QC, and Glyn, as Tim alluded to, will actually talk a lot more on the QC data that we have established to qualify these cells moving forward. Then, when the bank has actually been made of the cells, we actually transfer cells to our mirror facility at Fraunhofer IBMT in Germany to actually negate the sort of issues that, if one freezer goes down, then we have a backup, etc. And we ship to ECAC for distribution. So in the course of this project, where it's anticipated that we will be establishing 280 new FPIR cell lines, there will be cell line panels from diseased patients, and also with normal and sibling pairs who have disease or no disease. In addition, there is a number of gene-edited cell lines that we are establishing, and we have brought in a number of collections from Horizon 20 and IMI collaborations. So what we're building is this centralised activity in the EU for the collection, testing and distribution of these well-qualified IPS lines to under-standardised, diverse disease representative lines and panels which are reproducible and will give you a quality of cells to actually allow you to undertake your research. We've concentrated, because of the FPA partners' interest on most of the neurodegeneration and cardiovascular side. So we have representative lines in the catalogue from Huntington's, from Parkinson's, Alzheimer's disease, etc. And these will actually be expanding as the catalogue is built. So it is anticipated that we could, or you as a researcher, could use the IPS lines to stratify the disease or stratify based on drug response using these differentiated cells in a disease environment and looking at specific disease endpoints. So the current catalogue, and I've had to put this qualification up that it's as of the 18th of June, which is when I prepared this slide. I have had a deluge of emails to say that there's been a number of cells released. So this catalogue is actually changing, dynamically. So I would encourage you to go to the website to actually, to EBiSC.com to actually look at the catalogue as available, and it will be demonstrated in this session. But just to show the diversity of the lines that we have and that this will be building in the future. Also to note, specifically in a lot of these lines, there are 2 clones available in the catalogue, so you can actually build up a very comprehensive understanding of the disease etiology moving forward. So it's been a heroic effort across the whole of the consortia to actually really procure the IPSC supplying centres to get the information from the donors, the cell history, to expand, cryopreserve and quality control. And we're really, here now sort of talking about a very well-established process that would result in you being able to order a particular cell from a well-characterized cell bank to allow you to do the research. This gives a feel for the process steps that are involved in the expansion of the cells, and routinely we would have approximately 50 within a working cell bank, but the availability to go back to our master cell bank to generate passage similar passage numbers to the initial working cell bank if required when the cells have actually, the vials have been exhausted at ECAC. The QC this is a very general snapshot to show that we have cell phenotype, genetic identity, the pluripotent potential, chromosome stability and sterility of all the cell lines within the catalogue. And this actually shows a vial that is actually being distributed from ECAC with the code, what passage number, and it links it back to the data sheet that actually gives you all of the information on these cells. So we had these hot start lines, and we had 47 in total. And I just wanted to share with you how they performed during the process. So remember that this is 7 iPSC derivation centres. We actually had a training program so that all of the researchers supplying the cells into the bank were actually trained in all the processes that we wanted to adopt. So what media to use, how to passage them. This was actually a very well sort of regarded workshop, and is actually one of the highlights that people actually said in the whole process when it was initiated. So as you can see here, there were some individual results on the recovery of the cells, actually showing that there were 12.8% of the cells there were issues recovering the cells. They either didn't stick down or they didn't expand, they didn't perform as expected. There was quite a high percentage of spontaneous differentiation in these cells, but they were very clean with regards to viral screening, but a little bit of fungal contamination across these 47 lines. However, what was worrying, but what does agree with literature out there, was that 17% of the cells that we received in the central facility were actually not what we expected. So there was some mix-ups at the actual derivation center. So a female actually was a male in one instance. So, you know, this is something for other banking initiatives to be aware of, that you do need to look at your cell line identity quite carefully. But on the positive side, it should be N equals 38, not 38%. 38% of the cells, 38 of the cell lines that we received actually went on to 100% to go on to trilineage differentiation of forming mesoderm, endoderm, and ectoderm. So ECAC distribution, this is actually just the process showing that the vials are stored in a unique EBiSC freezer, so there's no contamination with other cell lines of unknown mycoplasma levels. They're easily traceable and they're shipped within the appropriate packaging. The examples that I've got are 3. So this is actually a slide from one of the partners, Definigen, who actually have a hepatocyte and also pancreatic cell differentiation protocol that we have been using to actually look at the ability of the iPSC lines to differentiate into hepatocytes and pancreatic cells. And this actually shows very nicely that the IPS lines that have been banked within EBiSC are actually have the ability to differentiate into very well characterized hepatocytes, as shown by the albumin levels and by the presence of the alpha-1 antitrypsin. So this is actually a key, metric to show that these cells have the ability to differentiate. This panel is actually from a Bioneer line, which is in the catalogue, showing that they differentiate to a neural lineage. The MAP2 are actually present on dendritic cells, and total tau showing that you've actually got a good network, and beta-3 tubulin as well. So this is an example from the University of Bonn, Oliver Brusel's lab, actually with a disease line. And I wanted to show you the actual differentiation capabilities of a control line on the right-hand side of the left-hand side of the right-hand panel. You've got the control line showing that you've got an increased neurite length, but in this hereditary spastic paraplegia, or HSP, you can actually see that there is a reduced neurite length in the iPSCs once they've been differentiated into neurons. And this is actually something that is being looked at in a drug screening campaign to see if you can recover this neurite length in the disease lines. So just showing that they have the ability to really give very good results in a differentiation capability. So just to say, I'm summarizing that we're actually building this centralized activity. We're at the first stage of really delivering the initial catalogue, and this is constantly changing, and we will be putting in a huge amount of IPS lines from specific diseases, and it's an opportunity for you to, if you have an interest in generating a particular line from a rare disease, then this is something, please come and talk to us. But we are, really, I think, delivering to a very high standard. So just to say, this is not a single entity, this is a consortia, and the next slide just shows the breadth of the individuals who have been involved in this. This is actually taken from the last year's general annual meeting, which was held in Granta Park, but we've had a more recent one in Berlin, and I'm sure that if you'd like to see the slides, we can actually present you with them. So thank you very much for your attention, and I'll take any questions.